What we know is chemotherapy has very debilitating side effects, but we also know in terms of access to treatment for cancer, because a lot of these drugs are very expensive, so there's not equal access across the world. So this means that outcomes for patients are very unequal. So we look at combating this using molecularly imprinted nanoparticles because they're much more cost effective. And also because we have developed a unique approach which can deliver these drugs to the nucleus, so only to the cancer cells. So when we started this study, you've seen now examples of how we can very selectively sense certain things, but when it comes to drug delivery, we show that we can not only detect the cell receptor, but we also have the ability to retain, so we also have high selectivity for the drug that we work with. So when we started this, there was only work done on the membrane receptor. So what we did is we can recognize the cell receptor, the particles move inside the cell, and there they can actually deliver the drug itself. And we adapted this to breast cancer. So we use the solid phase synthesis approach. So we use a very small, linear amino acid epitope, which is similar to the antibody that was developed for estrogen alpha receptor, and most breast cancers overexpress estrogen alpha. Then we collect only the high affinity particles, and we use this approach because we can actually recycle the material. So that makes it very interesting in order to recycle things and makes it more cheaper. What we showed is that we have a control cell line which is negative for estrogen alpha, and we saw with confocal microscopy that there was very selective binding only to the cells that were overexpressing estrogen alpha, and that also, if we compare this to the drug, that also there was a sustained release over time to these cells that were positive estrogen alpha. Whereas if you look at the right, there was hardly any cell death for those cell lines that didn't have this overexpression. One of the most interesting bits, so until now no one has actually shown that these particles can go into the cell, and via the confocal microscopy we can very nicely show that the efficacy of our approach was so high, because you can see over time, if you look at the little cartoon, these really penetrate into the nucleus, which is very unique to the system. Now what we're looking now at, and we'll show later on in the poster, is while we showed this for breast cancer, this can very easily be adapted to other cancers and to other drugs. So hopefully you've seen our polymers can enhance its efficacy, much more cost-effective solution, and we're looking at expanding this to other cell lines, and more importantly, we're also doing this work at the moment in animal models to show that they can very selectively also kill cancer cells instantly.